The Ediacaran period lasted a long time. The Ediacaran Biota assemblages ranged from about 575 million years up to about 540. They're not, as many often assume, a single continuous assemblage. There has long been a recognition of three distinct phases within the Ediacaran biotas. Now there's a new paper that suggests that this is not quite right, that possibly the first two were actually contemporaneous but in different environments, and there was a huge great mass extinction event at 550 million. So let's dive into this and see if we can get our heads around it. Hello everyone. Are you sitting comfortably? Because it's time for more Ediacaran stuff. This seems to be a really popular subject here, and I'm not surprised because it is a really fascinating time. Now, I am not an Ediacaran specialist. I don't actively research this, but it's something that's very close to my research interests, so I tend to follow it quite a lot. And whenever there's particularly a new fauna, a new biota, it tells us usually quite a lot more that we didn't recognize before, and this is one of those cases. So I'll give you a little bit of brief intro into the Ediacaran and the types of assemblages of fossils that we get. But if you would like to see quite a lot more detail about the fossil biotas that we get at this time, then have a look at the Cambrian explosion video that I tried to summarize how all the different types of fossils appear within this last part of the Precambrian, just before the main phase of the Cambrian explosion. So, Ediacaran fossils are really divided into two or three major groups. Firstly, there are the microfossils, which you dissolve out of cherts and limestones, and that's an entire field of its own, really. But it's mostly single-celled or filaments or lots of peculiar things that we don't know what to do with. But when we talk about macrofossils, there are really two very distinct types of preservation and fossil, which are more or less segregated. So on the one hand, we get macroalgal floras, which are basically carbonaceous compressions, usually in mudstones, sometimes with pyritization going on, where you have an assemblage of dominantly what are interpreted as algal fossils. And these have quite a long history, going back to about 580 million at least. And there are lots of deposits, although mainly in the region around China, north, south, Mongolia, that sort of area. There aren't so many of these deposits elsewhere. On the other hand, there are the classic Ediacara Biota fossils. And these are these really strange, weird preservation and weird morphology organisms that we get in a range of different sites around the world. Pretty much everywhere around the world there are elements of it. And the classic one that it's named after is the Ediacara Hills in South Australia, but there are equally famous sites now, the White Sea in Russia, the Mistaken Point, Newfoundland, and Charnwood Forest in England, the Nama beds in Namibia. There are a lot of deposits around the world. And this is what we're going to be focusing on today. Now, some people, including the authors of this recent paper, interpret pretty much all of these organisms as animals and mostly crown group animals, including bilaterians. And this is one school of thought which is currently very popular. I'm not going to go into whether that's right or not, because there is no answer on this yet. My personal view is that they're mostly stem group animals, probably independently multicellular from the familiar crown group animals, but we don't have a definite answer on this, and it doesn't matter for this particular video. Because what we're going into today is the basic patterns of diversity and abundance and morphology that we get within the Ediacara Biota assemblages. So since 2003, when Ben Wagner proposed this first, there has been an understanding of three successive Ediacaran Biota assemblages. Now, the first is from about 575 to 560 million years and is referred to as the Avalon assemblage, after the Avalon Peninsula in Newfoundland. This is the mistaken point biota, and there are multiple layers in this interval. And these assemblages are relatively limited. They tend to be dominated by rangiomorphs and some arboreomorphs. So there's a couple of major groups of essentially mostly frond-like organisms with a circular holdfast, some sort of little stalk, and then either branched or single branch structures with various patterns of branching of these sort of quilted cell structures, but not biological cell, but constrained units within these organisms' body plans. It's a relatively limited assemblage, and these are of extraordinary interest because they are the earliest examples. Now, there are other peculiar things within them. There are things like Aurora lumina, which has been interpreted as a cnidarian. This is from the Chanwood Forest site, so still about 560 million years. And it's a very interesting thing. There's just the one example of it. We'll do a whole video on this at some point to see what we can actually be certain of. But there is some diversity, is what I'm saying here. It's not just the classic fronds and candelabra type shape things. There's a little bit more to it than that, but it's still relatively limited. Then at about between 560 and 550, we get the absolutely classic Ediacara Biota assemblages. So these include the Ediacara Hills and the White Sea of Russia, where we have the most extraordinarily diverse and morphologically diversified groups of organisms. This includes things like the Dickinsoniids and Tribrachidium, Parvancorina, all of these things that some people now believe are bilaterian. Others are less convinced and are thinking that these are much more potentially allied with the earlier rangiomorphs, with a similar sort of body plan. This goes back to the Venda-Bionta idea of Seilacher. And again, the interpretation of them is not important today. The critical thing is that they are much more diversified in body plan. We're no longer looking at simple sort of frond or branching structures with a sort of stalk and a holdfast. Some of these were mobile. Some of them leave trace fossils behind them, it seems. Things like Kimberella, for example. Many of them are not stalked. They're sort of flat lying on the surface. It's a much more interesting and diverse community. And then that all changes. At 550 million, there is a transition into the Nama beds. Now, these are based on the Nama beds of Namibia, and you go from this extremely diversified assemblage to a much more depauperate one. The dominant groups now are ernietomorphs, which are weird sort of bag-like things you often get in 3 dimensions within sandstone beds, but often transported sandstone beds. They were long interpreted as being in situ, sort of mostly embedded in the sand. But it looks like now there is quite a lot of evidence that these are mostly transported. And it's difficult to work out from that what their original life habit was, whether they were embedded in the sediment or not. We're just not sure anymore, I think. And alongside them, we get the rise of tubes, organic, calcareous, all sorts of peculiar things, which are often assumed to be early animals, cnidarians and allies, possibly even in some cases annelid-like things. But there are also alternative interpretations of things like algae and various other groups. So there are tubes. We don't know what made many or all of them. And that is still mysterious in that sense, but it looks like it's getting more animal-like, in terms of more like the modern groups of animals that we are familiar with from the Cambrian onwards. Most of that assemblage then goes extinct at the end of the Ediacaran, at around 538 to 540, but the Nama beds are this unique assemblage in the latest part of the Ediacaran. There are some leftovers, hangers-on from the previous White Sea assemblage, so you do still get little fronds. And occasionally we get these even in more Burgess Shale type preservation, like in the Tongshan biota, where we have a dominantly algal frond-like assemblage, and then there are some of these Ediacaran vein-geomorph type things in there as well. So it's a really interesting suite of assemblages which appear to be distinct. The big problem is that we don't have large numbers of these fossil assemblages from different times all the way through the sequence. We have very few really good examples, and they tend to be restricted. So what we find is that the Avalon assemblages, these earliest ones, are all deep water. We do not have any shallow water equivalents which were living at the time of the Avalon assemblage, so between 560 and 575. There just isn't really anything known. So the deep water ones are often thought to be really quite deep, so mistaken point might be 600-700 meters, something like that. And this is potentially well below the photic zone, anoxic, in a very, well, at least very low oxygen. There's quite a lot of evidence that some of these faunas, there's one research group in particular that has been showing that a lot of these Ediacaran assemblages seem to have thrived in very low oxygen conditions. But it was a particular type of paleo environment. Now, the White Sea assemblages, what we have there are shallow water biotas, which are in sandstones on shelf deposits with microbial mats everywhere. And they're preserved as these weird preservational styles within microbial mat grounds. The Avalon assemblage ones, by the way, are mostly preserved under volcanic ash beds, with microbial mats on the surfaces because they were everywhere. But the volcanic ash beds seem to have been critical there, but we don't get them in the later deposits, so there's a taphonomic difference as well. But the White Sea assemblages are universally in this relatively shallow water environment. And that is a problem because we have these two successive assemblages, but one is in deep water and one's in shallow water. And we don't really have any way of matching up whether this is a temporal change, that there is a transition at 560 million years between a global ecosystem, which was all pretty similar, to a different one, or whether there were distinct ecologies developing in different environments, and they were all carrying on at the same time. We just don't see it. But then you get to the Nama beds, which are also very shallow water. And there we get a whole suite of new things like the tubes coming in, which you would imagine would be preserved in even slightly more offshore conditions in the White Sea assemblages. And we don't see such a contrast in paleo environment. So it looks like the Nama beds probably were ecologically distinct and represent the same sort of environment as the White Sea assemblage. So Nama beds look like there is a transition from the White Sea. But we have no idea whether the Avalon assemblage carries on even all the way through. Does it even carry on into the Cambrian? If we don't have equivalent ecology, we don't have equivalent sites preserving that environment, we can't tell. So this brings us to the latest paper. This is one by Duncan McIlroy et al., and it's based on work on the in Newfoundland, in the same area as the Avalon Peninsula, but a new site from a different part of the succession. They're still exposing the site. It is in a field, and they're having to dig the surface off. But what you find, like most of these deposits, is we have a single bedding plane. I think it's 40 square meters they have so far, but it's still ongoing. And it looks like it's going to be very extensive. This is a preliminary report. It doesn't go into all the detail. There are some new forms there, which is always good to see. And in terms of the basics, it appears to be very similar to the other bedding planes that you get at Mistaken Point. There are about 19 genera, they think, compared with 17, which is typical for the other sites. So very standard, similar preservation. There's a volcanic ash layer on the surface, and these things are preserved within microbial mat ground underneath the volcanic ash. So all perfect. Lovely. Now, the difference is that they've dated that volcanic ash, and they've come up with a very young age, comparatively. So the dating is fairly convincing. They got pristine zircons out of the volcanic ash layer. And the technique was, let me see, 6 single crystal zircon chemical abrasion isotope dilution thermal ionization mass spectrometry UPB analyses. Yeah, I think that's fairly convincing. So it's uranium-lead dating, basically, from 6 different zircons, and they end up with a very nice, tightly constrained date, actually. Error bar is about 0.6 million years, of just under 551 million. So this is not only well outside the Avalon assemblage range, which is 575 to 560, but it's right at the end of the White Sea phase. So this is one of the things we've been looking for, a deep water equivalent of the White Sea assemblage, and near the end of it, so near the top of that ecological assemblage. Okay, so what have they got? It turns out they've got pretty much a standard Avalon assemblage. It's got lots of rangeomorphs, it's got some rare arboreomorphs, there are a few newcomers, so there's a specimen of an ernietomorph, Philozoan. This is one of the group that dominates the soft-bodied stuff in the Nama beds, which is interesting. There are precursors of them, there are earlier ernietomorphs as well, so that's not a problem. And it's just interesting to see one of those turning up here. It again confirms this is likely a very young age, as shown by the zircons. And as I say, similar generic diversity to the other mistaken point biotas. So we've got something here which seems to be showing continuity of the deep water assemblage into much younger layers, which are equivalent to these really diverse White Sea assemblages in Ediacara and Russia. And what you get is a continuity of the deep water assemblages. Apparently there are only 6 genera from the classic mistaken point assemblage that they haven't found in this layer yet. So it is pretty much entirely continuous with those older beds. It looks like for the deep water there was no transition at 560 million, they just carried on going. It's just that we haven't seen them anywhere else. Okay, that's really interesting. It suggests, if this is standard, if this is a representative example of the rest of the world, it suggests that the White Sea assemblage was evolving, or had evolved, in shallow water environments, and that was what it was adapted to. And at the same time, the deep water environment was hosting a slightly more limited, but perhaps longer lived, because we still don't have any early shallow water faunas. If we had a shallow water Ediacara Biota assemblage from, say, 560 or 570 million years, maybe it would show that these classic White Sea assemblages actually carry on a lot further back. But we don't have that. We do have things like the Lancienne biota, though, which are these algal-dominated assemblages. And in the latest Ediacaran, the Tongshan, we do see some fronds preserved in that setting as well. So you might use that as at least weak evidence, given that we don't see anything like that in the oldest ones, that the shallow water assemblage hadn't fully evolved at this point. And it looks like there was potentially a deep water origin of the groups in general, the rangeomorphs, arboreomorphs, and so on, during the time of the Avalon assemblage, before you had diversification in the shallow water where they could be preserved along with the algae. So it does look like, from what we've got here, that there is still an earlier origin in deep water and then a later diversification in shallow water, but we don't know for sure because we don't have the continuity of the preservation. But it does look like now, at least, that there is some major event at about 550 million years, which has been known as the Kotlin Crisis. And this new paper is basically saying that this was the first major mass extinction event, of animals at least. As I say, these authors are very much in the camp of these all being metazoans, and usually Cran group metazoans as far as I can make out. That's fine. It is definitely a valid point of view. But so they're saying that this is the first mass extinction experienced by animals. And interesting. The problem is that this is based on this extension of the Avalon assemblage into younger rocks where they also go extinct at 550 million. But we don't have any evidence of this deep water assemblage after 550. And the problem is we don't even have the potential for that because we don't have those ecosystems preserved. Above this, in the Avalon area, it goes into deltaic sequences, which are much more, probably much shallower water, but much more turbulent, mobile sediment, just not suitable for the same community. And you do get a much more limited assemblage of little discs and things like that at that point. So what we see there isn't actually able to answer the question of whether the deep water assemblages carried on in that area even. So we don't know for sure that the Avalon assemblage actually went extinct at this 550 million marker, the Kotlin Crisis. We also don't know at this stage whether this is a geographic thing, because our Avalon assemblages, the deposits, are all mostly reasonably high latitudes. So we don't know whether this was a high latitude effect or whether it might have been something that was truly global. We also, as I say, are still not sure about whether there is a real ecological difference in terms of the paleo environment between the White Sea and the Nama assemblages, but that looks less convincing. There's quite a lot of work now suggesting that actually these two assemblages are pretty much interchangeable in terms of the paleo-environmental type. It was all sort of shallow water, sandy environments, and with a lot of similar sort of depth indicators from the sedimentology. So there certainly was a crisis there, and we don't know whether it affected the deep water, but given that we don't really see these things turning up elsewhere, we have to assume that it did, but it is an assumption. So what could have caused this crisis? Well, there is some discussion in the paper, but it's a little bit sort of vague. It seems that there aren't any obvious triggers. One thing they discuss is the rise of predation, but then in their interpretation of many of these as sort of Cran group animals, they also totally accept that cnidarians go back much, much further. So you have predation already in this ecosystem. And perhaps in that case, they're suggesting that maybe cnidarian predation contributed to the demise of the sessile rangiomorphs and arboriomorphs a bit. But it's difficult to see that coinciding with a sharp event, which actually knocks out most of them. It's also worth noting on the idea of cnidarian predation that cnidarians are microcarnivores. They feed using nidi, these little harpoon-like cells which fire when they're in contact with something. And so they're not going to be going around eating large rangiomorphs or anything like that. These are going to be feeding on potentially microscopic dispersal larval type phases. That might have a significant impact. But again, most of these will be probably feeding on very small organisms. So it's difficult to see that being a really major factor for me. They also then discuss the role of burrowing, increased bioturbation, particularly in affecting things that were upright, relying on a holdfast on a mat surface, and you start disturbing that mat and things start falling over. So yeah, that is definitely a factor. But again, that you'd think would happen later on during the Nama assemblages, and because the trace fossils increase steadily through that latest part of the Ediacaran. So again, it doesn't seem to tie in with a sharp event at 550 million. And it doesn't appear at the moment that there is an obvious reason for this crisis at that time. There's one other major candidate, which is something like an anoxic event, where you have shifts in oxygen levels And there is a lot going on in terms of the geochemistry of the oceans at this time. There are spikes all over the place, so that is entirely plausible. But there is, as I say, quite a lot of evidence that at least some of these organisms were adapted to very low oxygen conditions. And especially, you would think, the deep water assemblages. So if that was the case, if it was an anoxic event that caused this crisis, then you would expect the least affected organisms to be the ones that were adapted to such environments and were living in the deep water. So maybe this Avalon assemblage actually did carry on quite a lot longer. We don't really know. Because, yet again, we don't have the evidence for it at this stage. So this is very much a preliminary paper. It's announcing the discovery of this Inner Meadow site, as they call it, the continuation of the Avalon assemblage up into much younger rocks. And that in itself is really interesting and actually quite a major discovery. And then expanding that to extrapolation of the first Eumetazoan extinction event, mass extinction, which, okay, that part's debatable to me, but it is certainly looking like there was a crisis there. If, however, this deep water assemblage carried on in the deep water, but we're just not seeing it, then we don't know that this crisis is actually any more dramatic now than it was before this site was discovered. But it is really interesting, and it is part of this evolving tapestry of fossil assemblages, which seem to be more or less global within a particular environmental range. And we have to say more or less because we don't have a global coverage yet. We have a lot of sort of isolated little bits with aspidella discs and things like that in quite a few places, but good assemblages are quite few and far between. So there's a long way to go yet in understanding exactly how these little communities were evolving and how they were distributed around the world. But this is a big step forward. It's really interesting to see, and hopefully there'll be more coming out of it. And there will be a lot more coming out here as well. So when we get new discoveries, of course, we're going to be covering them, and we'll see where it all leads. But there's no definite answer for this one yet, I think, beyond the fact that these deep water Avalon assemblages did carry on a lot later than we thought. So there we go. I'll leave it there. It's published in Geology, so I'm afraid it's not open access. I got a copy from the authors, and it was well worth it. It's a lovely new assemblage. Right, we'll see what we can do next. As I say, I'm running a little bit sort of short of time at the moment because of other work that I'm doing on the side, and I have been asked by quite a lot of people to do something about what I'm working on with the modern mites and springtails and these soil samples, so I will cover that as well. But thank you very much for your interest, and I'll look forward to seeing you again. Bye for now.